In this lecture, we will study about enamel, its properties and structure. The objectives include understanding the properties and structure of enamel. Enamel, the hardest biological tissue, forms the outermost layer of the dental crown. Originating from epithelial cells, it is created by ameloblasts and cannot regenerate once damaged. As a non-vital tissue, enamel is acellular and avascular post-formation, making its preservation crucial for dental health. Enamel covers the entire crown surface of teeth, with thickness varying significantly. It is thickest on the cusps of molars and premolars, measuring 2.5 millimeters, and thins to a knife edge at the cervical region. Notably, maxillary molars have thicker enamel on lingual surfaces, while mandibular molars are thicker on buccal surfaces. Enamel's specific gravity is 2.8, indicating its density surpasses other tissues. This density diminishes from the surface inward and from cuspal to cervical areas. This value represents the ratio of the density of the enamel to the density of water, making it a dimensionless quantity. Functioning as a semi-permeable membrane, enamel permits certain molecules, like carbon-14 labeled urea and iodine, to pass through its structure. Enamel color ranges from yellowish-white to grayish-white. A yellowish appearance occurs when thin, translucent enamel reveals the yellow dentin beneath. Conversely, a grayish appearance suggests a more opaque enamel structure, affecting its translucency and overall aesthetic. Enamel translucency is influenced by calcification degree, where higher calcification results in opacity. Structural homogeneity enhances translucency, while light transmission is affected by enamel thickness and mineral content. Additionally, hydration status plays a role. Dehydrated enamel appears more opaque compared to hydrated enamel. This table summarizes physical characteristics of enamel. Enamel is composed of 96% inorganic material, primarily hydroxyapatite crystals, and 4% organic substances and water. The organic component includes unique proteins. 90% are hydrophobic amylogenins, rich in proline, histidine, glutamine, and leucine, while 10% are non-amylogenins like enamelin, ameloblastin, and tuftelin, rich in glycine, aspartic acid, and serine. Enamel's structure includes rods, rod sheaths, and interprismatic substances. Rods, cylindrical and wavy, extend from the dentino-enamel junction to the tooth surface. Key features include Hunter-Schreger bands, incremental lines of Retzius, and surface structures like enamel cuticle. Hypocalcified areas involve rod sheaths, enamel lamellae, and neonatal lines, impacting enamel integrity. Enamel is structured with rods, rod sheaths, and interprismatic substance. Enamel rods, cylindrical in shape, extend from the dentino-enamel junction to the tooth surface. Lower lateral incisors contain approximately 5 million rods, while upper first molars have around 12 million. These rods follow a wavy path, especially elongated in the cusp region. Alternating deviation of enamel rods from the radial direction is evident throughout the crown. This deviation contributes to the enamel's resilience and strength, allowing it to withstand various forces. The rod's orientation changes, enhancing the tooth's ability to resist wear and fracture, crucial for maintaining dental health. Rod length exceeds enamel thickness due to their oblique orientation. The diameter increases from the dentino-enamel junction to the surface, approximately doubling. Cross-sections show rods as hexagonal, round, or oval, resembling fish scales. 3D imaging reveals arcade-shaped rods near the junction, and keyhole-shaped rods at the enamel surface. Electron microscopy reveals that enamel rods are keyhole or paddle-shaped prisms, measuring 9 micrometers in length and 5 micrometers in breadth. The bodies of these rods are located near the occlusal or incisal surface, while the tails point cervically, providing structural integrity and functional adaptation in dental enamel. Electron microscopy reveals that human enamel features keyhole or paddle-shaped prisms. These rods are encased by rod sheaths and interrod substance. Longitudinal sections intersect the heads or bodies of one row of rods and the tails of an adjacent row, illustrating the intricate structural organization. The thin peripheral layer is rod sheath and is less calcified. Between rods, interprismatic or interrod substance is present. Hydroxyapatite crystals are arranged nearly parallel to the long axis of enamel rods. These crystals measure between 0.05 to 1 micrometer in length and 90 micrometers in width, adopting a pyramid shape. This structural arrangement is crucial for the mechanical properties of enamel, influencing its strength and resilience. Polarized light and X-ray diffraction studies show that apatite crystals align parallel to the long axis of enamel prisms. This precise orientation is crucial for the structural integrity and function of enamel, ensuring its resilience and durability. In the body and head of enamel rods, crystals align parallel to the long axis. However, in the tail of enamel rods, these crystals deviate approximately 65 degrees from the axis, indicating a distinct structural variation within the enamel rod itself. Enamel rods and teeth are oriented at right angles to the dentinal surface. In deciduous teeth, rods are horizontal in the cervical and central crown areas, becoming oblique near the incisal edge. In permanent teeth, this pattern is similar in the occlusal 2/3, with cervical rods deviating apically. Picture showing direction of enamel rods in deciduous and permanent teeth. Gnarled enamel appears near the dentin and cusp or incisal edge regions, where bundles of rods intertwine irregularly, especially in oblique sections. This optical appearance is significant in understanding enamel's structural complexity and its role in enhancing durability in high-stress areas. Gnarled enamel features an interwoven, twisted arrangement of enamel rods, primarily in cusp areas. This formation arises from the crowding of amyloblasts during enamel matrix deposition in confined spaces. Clinically, it enhances strength and wear resistance, bolstering structural integrity in high-stress regions. Hunter-Schrager bands are alternating dark and light bands in enamel, visible in longitudinal ground sections under oblique reflected light. They result from periodic changes in enamel rod direction, enhancing enamel strength and reducing fracture risk under occlusal forces. Hunter-Schrager bands consist of diazones, the dark bands formed by longitudinally cut prisms, and parazones, the light bands from transversely cut prisms. These bands, originating at the dentino-enamel junction, extend outward with an angle of approximately 40 degrees between them, contributing to enamel's structural integrity. Incremental lines in enamel arise from rhythmic deposition, with mineralization following matrix patterns. Hesitation during deposition leads to mineral content variations. Spaces between enamel crystals trap air, enhancing line visibility. The striae of Retzius, first described by Dr. Retzius, are key growth lines reflecting these processes. The neonatal line is a distinct stria of Retzius marking birth. It highlights the transition from prenatal to postnatal enamel, reflecting abrupt environmental and nutritional changes. Prenatal enamel is smoother with fewer defects, while postnatal enamel has more spaces and stains more easily, indicating the impact of birth on enamel formation. Figure showing neonatal line and lines of Retzius. The structureless enamel layer, approximately 30 micrometers thick, is present in 70% of permanent teeth and all deciduous teeth. It is commonly found in cervical areas and is least present at cusp tips. This layer lacks prism outlines, consists of parallel apatite crystals, and is more mineralized than the underlying enamel. Parachemata are transverse, wave-like grooves on enamel, representing the external manifestation of the striae of Retzius. They run parallel to the cemento-enamel junction. Density varies, with approximately 30 per millimeter near the cemento-enamel junction and about 10 per millimeter near the occlusal or incisal edge. Picture showing parachemata. Bottom right figure is electron microscopic appearance. Ultrastructural features of enamel include pits, representing ameloblast ends, and enamel caps formed by deposition on non-mineralizable debris. Enamel brocks are larger elevations on the surface. These features enhance enamel strength, wear resistance, and play a crucial role in mastication and maintaining structural integrity. Enamel lamellae are thin, leaf-like structures extending from the enamel surface toward the dentino-enamel junction, sometimes penetrating into dentin. They are rich in organic material and have low mineral content, often confused with cracks in ground sections. Formation occurs due to planes of tension and incomplete rod calcification. Enamel lamellae are categorized into three types. Type A, which consists of poorly calcified rod segments. Type B, filled with degenerated cells. And Type C, containing organic material from saliva. Clinically, these lamellae can act as weak points in enamel, potentially serving as pathways for bacteria, thereby increasing the risk of dental caries. Previously known as cracks, these are now recognized as the outer edges of enamel lamellae. Typically less than 1 millimeter in length, some extend to the occlusal or incisal edge. They appear evenly spaced, with longer lamellae being thicker, and run at right angles to the dentino-enamel junction. Enamel cracks can serve as pathways for fluids and bacteria, potentially leading to enamel fractures and dental decay. Understanding these pathways is crucial for preventing further damage and maintaining dental health. Nasmyth's membrane, or the primary enamel cuticle, is a delicate basal lamina secreted by ameloblasts post-enamel formation. It covers the entire crown of newly erupted teeth, protecting enamel from resorptive activity before eruption. Typically, it is removed by mastication. Nasmyth's membrane is a delicate basal lamina secreted by ameloblasts after enamel formation. It covers the entire crown of newly erupted teeth, protecting enamel from resorptive activity before eruption. However, it is likely removed by mastication, leaving the enamel vulnerable to external factors. The acquired enamel pellicle is a thin protein layer from saliva, forming within hours after cleaning. It acts as a protective barrier. However, within 1 to 2 days, microorganisms colonize it, leading to biofilm and potential dental plaque. While it protects enamel, it can harbor bacteria, increasing the risk of dental caries. Hypocalcified ribbon-like structures, known as enamel tufts, extend from the dentino-enamel junction into the enamel. Resembling tufts of grass under low magnification, they are rich in organic material, comprising hypocalcified enamel rods and interprismatic substance. These structures develop due to spatial constraints during enamel formation, extending 1/5 to 1/3 into the enamel. Enamel tufts align with the crown's long axis, often visible in transverse ground sections. These are hypomineralized regions, and a 13.17 kilodalton protein called tuftalin is present. Odontoblast processes extend across the dentino-enamel junction into enamel, forming spindle-like structures. These spindles, thickened at the ends, align at right angles to the dentin surface. They appear dark in ground sections due to organic material disintegration. These are hypomineralized structures and are predominantly located in cusp tip regions. They may enhance enamel resilience and crack resistance, contributing to the tooth's structural integrity. Age changes in enamel include loss of enamel, called attrition, due to mastication, losing pericomata post-eruption. Fluoride uptake increases in superficial layers, reducing permeability. Enamel's brittleness makes it vulnerable in thin layers or unsupported areas. Deep fissures and dental lamellae, rich in organic material, predispose teeth to caries, highlighting the importance of structural integrity for dental health. MCQs, feedback link, references.